I'm going to explain this. Remember I said to you there would be a second part to your lab, the NCBI. You know how you're building your trees? And I said it would be second part. I was working on the second part because it takes a long time to go through and find the right protein to use. So I came across the proteins. I'm going to tell you now. I checked them all out. They should all work. I'm going to tell you what we're going to do with them. Okay? So what we're going to do is we're going to look at something pretty interesting. We're going to look at reconstructing what I'm going to call the last common ancestor of eukaryotes. Now, you guys know, or you may know, that there are eukaryotes, cells with a nucleus, and then there are prokaryotes, cells without a nucleus. I don't know if you know that. I don't know if you know this, but prokaryotes is another word for bacteria. And there are two types of bacteria. Okay? The two types of bacteria, and we'll look at this when we go into classification. But the two types of bacteria are archaea and regular bacteria, also called eubacteria. So you can spell it with the E, U, and then bacteria, or just call it bacteria. So there's two types of prokaryotes. There's archaea and there's eubacteria. Okay? And we'll talk more about that when we get to the whole section on classification. So all you need to know right now is that there's different types. Here's the thing. What do you think came first? Prokaryotes or eukaryotes? Yeah. Why? Right. I mean, we know from the fossil record and other forms of evidence, but just by logic alone, what's simpler? Bacteria or eukaryotes? So you would think something that's simpler would be easier to evolve than something that's more complicated. And that's true. Okay? So prokaryotes came first. Now the question we're going to ask is, okay, eukaryotes came after prokaryotes. Probably evolved from prokaryotes. And if we did evolve from prokaryotes, are we more like the archaea? Or are we more like the bacteria? Or are we a combination of both of them? Okay? That's what we're going to look at. So how many of you know what a chimera is? You've never... I bet you if you go to Google and you ask for a chimera, you might get maybe some images, something like this. What do you think a chimera is? So here, obviously, this is something that does not exist. It's mythical. But what do you think a chimera means? But it's a mixture. Okay? So a chimera here is a mixture of, in this case, 3 different things, right? So the question we're going to ask is, are we more like archaea? Are we more like bacteria? Or are we a chimera where we're a combination of the two? Okay? So you have here 10 proteins. What I've done to make it easier for you is I've given you the accession number, which is the identifier. What you can do is you can copy this and you can put it in an NCBI. Okay? So each of these, so here's a description of the protein. We're going to use 2 different organisms because I was having a hard time finding the protein in humans. So I said, you know what, we're going to use humans and fruit fly to make it a little bit easier. Because we're both eukaryotes, we're both animals. So, you know, we're connected, right? So I have stuff from fruit flies and stuff from humans. So I'm going to show you an example of what we're going to do, okay? So I have a protein here. It's not one of your 10, okay? So you don't have to worry about me doing the work for you. I know you don't like it when I do your work, right? Yeah, not at all. So the protein here is called, well, this is part of a protein. You'll learn about this protein in grade 12. It's involved in making energy, okay? This protein comes from Drosophila. The word isoform just means different types, okay? And this is actually a subunit or a part of this protein called ATP synthase, okay? Which is a protein that makes ATP. It comes from Drosophila melanogaster, which is a fruit fly. This here is the breakdown of the classification. So you can see here that it's a eukaryote. Okay? What we're going to do is we're going to run what's called a blast. Now, you guys have a little bit of experience with this. But we're going to do something a little bit different. We're not going to align the sequences. You know how you guys copied and pasted and you align different sequences? We're going to do something different, okay? So this is the code of that protein. Now, I provided you the code, so all you have to do is really copy and paste the code. So, for example, if we copied that code and we went to ncbi, you should just be able to paste it in, and you should get your protein. Okay? So you should get it back, and you can see it's right here. There's the protein. I should be able to click on that and get my protein. So you have the codes for you, so you don't have to worry about, because so what happens if you type in the name of a protein, you might get like 100 hits, and you'd be like, oh, which one do I pick, right? Right. Well, in this case, you don't have to worry about that because I give you the actual code, but that's the one you're going to use, all right? So what we're going to do is we're going to run a blast. Yep. Okay, so we're going to run a blast. But I'm going to do something different. What I'm going to do is I'm going to show you exactly what I did, okay? Remember how BLAST works, right? It takes a sequence and it compares it to all the other sequences in the database to find matches. What we want to know is if eukaryotes are more like bacteria or archaea, and they're prokaryotes. If we just say to the computer, go ahead and take this sequence and look for similar sequences, it's going to retrieve the ones that are most similar, which means it's going to retrieve the ones from eukaryotes and prokaryotes. So what's going to happen is you're going to get a hit, you're going to get a bunch of hits. The ones that are going to come up first are the ones that are probably animals or other eukaryotes. We don't want eukaryotes in our database. We want to compare it to bacteria. So this is what we're going to do. We're going to exclude, we're going to remove eukaryotes. Okay? So eukaryota. We're going to exclude them. So sometimes what happens, I notice that it doesn't work, but it should. Where you exclude something and it still pops up in the list, but it shouldn't, okay? So right now what I've done is I've basically said to the computer, okay, I want you to search, but if you come across a eukaryote, ignore that one. I just want things that are not eukaryotes, which means bacteria. But you might also get viruses in your list, because guess what viruses are? They're not eukaryotes. So you might get viruses. The database also includes models and some other samples. So I'm going to check off all these three boxes, and hopefully that will refine the search so that the only thing that we get are bacteria. Okay? So I'm going to run a blast now, but you know that it might take a while. So I've already run the blast for you. So I'm going to click blast, and you're going to see something like this, right? But I've already done it for you. This is it. This is the results from, in this case, you can see ATP synthase, okay, from the fruit fly. This is what comes up. It goes in order of similarity. So, like for example, this is 100% identical, and then it works its way down. What I don't want, because the computer is comparing it to everything in the database, some people have put in stuff in the database that are synthetic, things that have been created in the lab. That's not a living thing. So, for example, if I click on this, we'll see where it says synthetic. If I click on that, it takes me to this. This lines up the sequence of what I got with what I'm comparing it to. This is what it is. If I click on that, I'll notice it says organism. It says synthetic. It says artificial. So that is no good to me. What I want is I want things that are not synthetic. So I'm going to go across my list, and I'm going to say, okay, that says synthetic. Now this also says synthetic, but this doesn't. Okay, so what is this? So I'm going to click on that. Okay, here it is. I'm going to click on this thing and to see what it is. Okay, so here it is. Azospirillum halopreferens, I think. That's how you pronounce it. And here's my organism, and here is the classification. It comes under bacteria, not archaea. So for this protein, the first hit was bacteria. What I'm going to do, though, is I'm going to look at the top 3 that are non-synthetic. Okay? Or the other word that you might see is hypothetical. So if you see hypothetical or synthetic, ignore that. Okay? So I see this one, and I see another organism here. So I can click on that, see what that one is, see if that's also bacteria. See if it comes up. Okay? Here's a different specimen, and it comes up as bacteria also. What I want to know is, what are my top 3 organisms that come up in the hit? Are they bacteria or are they archaea? You're going to do that for 10 proteins, okay? So you have 10 here. You have DNA primase all the way down to DNA polymerase, okay? You have the accession code, so all you got to do is copy and paste that. You're going to run the blast. You're going to exclude the prokaryotes, and you're going to see what are your top 3 non-synthetic, non-hypothetical organisms. And you're going to record. So what you're going to do is you're going to provide a a table of your results. This is a protein. These are the top 3 organisms. What are they? Archaea or bacteria? For all 10. And then you're going to give me a really brief 1-sentence description of what these things are. And then what is your analysis? Are we more closely related to archaea or bacteria or both? And then I want you to research the endosymbiotic theory of the origin of eukaryotes. And how does that theory coincide with what you discovered? So in other words, if you discover that we are more like bacteria, because all of the hits come back bacteria, and that theory says we came from bacteria and not archaea, then you would say, well, the results match. Okay? By the way, I'm not saying that's the answer. I'm just giving you an example. Okay? So this is part B. Now, this part and the other part are due anytime next week. Okay? And I'm going to stop now. So you have the rest of the class if you want to start looking at this. Okay, ladies? That is it.